Malaria is a life-threatening disease caused by Plasmodium parasites, which are transmitted to humans through the bites of infected female Anopheles mosquitoes. Manual microscopy remains the gold standard for diagnosis. Types of malaria, Plasmodium species. Human malaria is primarily caused by 5 distinct species of the Plasmodium parasite. Each has unique geographical distributions and clinical impacts. 1. Plasmodium falciparum, the most dangerous and prevalent species, particularly in Africa. It can cause severe, life-threatening malaria and death within 24 hours if left untreated. 2. Plasmodium vivax, widely distributed outside of Africa. It is known for its ability to form dormant hypnozoites in the liver, which can cause relapses months or years later. 3. Plasmodium ovale, similar to P. vivax. It can cause relapses and is primarily found in sub-Saharan Africa and the Western Pacific. 4. Plasmodium malariae, known for causing a quartan fever occurring every 3 days, and can persist in the blood for decades if untreated. 5. Plasmodium knowlesi, a zoonotic species primarily found in Southeast Asia, transmitted from macaques to humans. It can rapidly progress to severe disease. Staining techniques and microscopy. Effective diagnosis depends on visualizing the parasites within red blood cells, RBCs, using specific stains that highlight parasitic DNA and structures. Gold standard staining. Giemsa stain, the most widely used technique for malaria diagnosis. It is a type of Romanowski stain that typically uses a 10% buffer diluted solution. It provides excellent contrast, staining the parasite's nucleus red/purple and the cytoplasm blue, making it easier to distinguish species. Types of blood smears. 1. Thick blood smear, used for detection. The RBCs are lysed, burst, allowing for a larger volume of blood to be screened, which is essential for identifying low parasite densities. 2. Thin blood smear, used for species identification. The RBCs remain intact, allowing clinicians to observe the specific morphology of the parasite and how it affects the host cell, e.g., cell enlargement in P. vivax. 3. Modern diagnostic alternatives. While microscopy is the clinical baseline, other methods are used based on the setting. 3.1 Rapid diagnostic tests, RDTs. Immunochromatographic tests that detect specific malaria antigens, like HRP2 or LDH. They are fast and require less training, but are generally less sensitive than expert microscopy. 3.2 Molecular testing, PCR. Highly sensitive and capable of identifying drug-resistant markers, but often limited to reference laboratories due to cost and complexity. 3.3 AI automated microscopy. New platforms, like MeLab, use deep learning and digital imaging to automate the staining and scanning process, achieving over 90% concordance with expert human microscopists.